Good morning. It has been a pleasure to come here, Egypt again. Every time I come to Cairo, it has changed. And changed much bigger this time than I saw last time. Before I talk, I'd like to thank Professor Abdallah for inviting me here to give a talk. It's always a pleasure to come to Cairo. I'll also like to congratulate her for organizing such a good meeting. Because it enhanced longevity, because I started working polyamine for different reasons long ago, because polyamine has been shown to play a role in plant senescence. So I thought I wanted to see what can I do by genetically engineering polyamine. But recently evidence began to point out that it has a lot bigger role than just inhibiting senescence in plants. These are the responses of polyamine in plants. There are multitude of responses in plants, and you can see it is involved in cell division, it is involved in metabolism, homeostasis, signal transduction, transcription, translation, stress response, totipotency, rejuvenation, crosstalk. And I do not know which plays role to what, but there's so many different phenotypic effects of polyamines that it has involved in embryogenesis, root development, flower initiation, pollen tube growth, list goes on and on and on. So keeping this in mind, we thought we'll change the metabolism of polyamines in tomato fruit. We understand very clearly the metabolic pathway for polyamine biosynthesis. It's made from two amino acids, arginine or orthonine, by enzyme orthonine decarboxylase or arginine decarboxylase. And then this putrescine, by using enzyme spermidine synthase and a decarboxylated SAM makes spermidine, and one more decarboxylated SAM makes spermine. I also remind you here, in plants, SAM is an important precursor for a plant hormone called ethylene. So, my medication kills me somewhere. Okay. So, what we've done actually in my lab, we tried to modify two enzymes. One is SAM decarboxylase, other is spermidine synthase, and I show you some phenotypes. One thing we find that when you express the SAM decarboxylase in a ripening tomato fruit, the level of polyamines go up in ripening fruit. For example, this is the putrescine, which goes down in both, which goes up in ripening fruit, but spermidine and spermine both go down. However, when you express the gene, you can get much higher level of polyamines. What they do, they basically make fruit much redder. And red color is coming basically from a compound called lycopene. And we find that even the tight regulation of lycopene can be changed by expressing SAM decarboxylase. So polyamines play an important role in production of lycopene, which has been shown to cause reduction in various cancers, including cardiovascular diseases. I do this a lot of work with one of my colleagues, Karmatu, in Beltsville. Then one of my students, Savitri, she basically looked at the other enzyme, spermidine synthase, and she expressed that in tomato fruit also. But unlike SAM decarboxylase, which has a lot of 2 to 3-fold increase in polyamines, there are not that much increase in this compound when you express spermidine synthase. It basically suggests that SAM decarboxylase is a rate limiting steps. But again, there are profound effect on shelf life of fruits actually. What we found that these fruits, which normally shrivel because of post-harvest storage, the shriveling was much reduced in these fruits. And you can see the tomato fruit shriveling time increased by 8 days more. So you have a longer shelf life. So you can sell the fruit for long distances actually. Most of crops shrivel because they lose water. But what we find here that it's not water loss. Water loss is the same in all of them, but still fruit stayed very nice looking, healthy fruit. So basically what polyamine is doing is maintaining the integrity of fruit and keeping it in more rejuvenated forms. It has very little effect on, it has some effect on post-harvest decay, but a couple of days is not very that significant. However, it changed ripening a little bit. Ripening was delayed somewhat in these fruits. And what we find that this ripening was not because of ethylene reduction. These transgenic fruit produced a lot more ethylene. in spite of a lot more ethylene, they still did not undergo decay. So it suggests that that 2 points here. One thing is that ethylene is not the only one which causes the senescence of fruit, and number two, it also suggests that SAM is not rate limiting step for ethylene biosynthesis. Both steps can exist side by side. It also increases the respiration process in fruits, and what we find the enzyme involved in the decarboxylase or acetate dehydrogenase, they all gone up enormously in these transgenic fruits. I'm coming to this why I'm doing this thing, all the thing, because I'm just showing you some phenotypes. It also changed. We looked at metabolic studies and changed some of the amino acids. Some have gone up. For example, glutamine has gone up, asparagine has gone up, whereas valine has gone down, and aspartic acid has gone down. Same thing was true for the sugars. Some of the sugar citrate has gone up, and some other choline, for example, gone up. And choline is very important compound for human health also. And based on data, what we suggest that basically polymer might be acting as a nitrogenous metabolite that stimulates carbon sequestration. Okay. And most interesting thing is that polyamine enhance the respiration and other processes in late ripening stage when it's supposed to be senescing actually. Other aspect, we looked at seasonal senescence of plants. And what we find that this basically shows that different days after planting, seed planting, this is wild type. Wild type begins senescence after 110 days in the field, where transgenic flower plants do not show much senescence and they maintain the vegetative weight. That's showing that you can extend the season for longer time if you have polyamine expression there. We make some microarray in our lab and we test the microarray, and what we find, there are a lot more genes which are upregulated than downregulated by the transgene. So basically it suggests that that polyamine has a stimulating effect on gene expression. And these genes were represented a lot of different types of metabolic pathways. You can talk about gene involved ethylene action, ethylene synthesis, signal transduction, nitrogen assimilation, protein biosynthesis. So collectively based on this data, we publish a proposal model that polyamine will store and enhance metabolic memory even in the late ripening stages. Okay. However, this model did not allow us to distinguish between the role of different polyamines. So what we did actually, so we proposed to test the hypothesis, all polyamine, this is our hypothesis, all polyamine act the same way and can substitute for each other action. It's like a ANOVA, null hypothesis. That's what we thought earlier. To test the hypothesis, we determine the correlation coefficient between endogenous levels of each polyamines and each of the parameters we quantified. And that's what you see actually. These are popular cell cultures, and these are tomato fruit. In cases of putrescine, you see it's at a negative. Most of the time, it reduces the level of all these metabolites, alanine, glutamine. Whereas in the case of spermidine and spermidine, they are upregulated. That's suggesting that it has a more anabolic function or a catabolic function, like not like the putrescine. Same was true for the when you look for the different transcript, and you find the lot of transcripts basically are downregulated by putrescine, while they are upregulated by spermidine and spermine. Not upregulated, they show positive correlation with spermidine and spermine. Okay. Same was true when we looked for some of the proteins. We did a lot of Western blots and find same thing. that proteins, there are more proteins downregulated in putrescine, showed negative correlation with putrescine, but positive correlation with spermidine and spermine. It's a quick slide because it's not that important. It's basically what you look at, that how much is down and how much is up actually. Similarly, we looked at the physiological parameter like ethylene production, like pH, Brix, acidity, viscosity, and it seemed like in all cases putrescine has a negative correlation compared to spermidine and spermine. So this basically let us suggest another paper we published in, I think it's an amino acid probably, yeah. The different polyamine, for the first time we showed that different polyamine have different roles. And putrescine acts as a negative regulator that initiates several catabolic responses, and spermidine and spermine act as a positive regulator which stimulates a lot of anabolic responses. Polyamine also has a morphogenic roles, and what we find the fruit's shape has changed. This is wild type fruit, and there are 3 different transgenic lines, and you can say it become very elongated one. And in this case, what happened that pericarp increased, and we have studied the role of cell wall, studied the role of the cell cycle, and what we find the gene involved in cell divisions as well as endoreduplication are highly expressed in high polyamine lines. So basically, based on all the studies, it seems like polyamine don't have a single mode of action. It acts on multiple stages in development. They act at the time of chromatin, they modify transcription, they modify translation, they modify protein structure and function, they modify the metabolites, and they also ultimately reflect itself in form of phenotypes, which is very useful. Okay, what we're doing now basically, we're taking tomatoes and trying to see if we can feed that to some animals like mice, and can we make them live longer. And 6 months from now, hopefully we'll know the results and see whether endogenous polyamines can really have a role in mammalian system to increase their longevity. Just few small, last 2 minutes I take showing some other interesting phenotype from my lab. One of them is basically, long ago we modified pectin chemistry, and what happened that this is a disorder for tomato fruit, they got blossom end rot because of lack of calcium. But since we changed pectin chemistry, these plants show very low level of blossom end rot, and that basically because the calcium level has gone up in apoplast. And this problem is around 20% loss in crop productivity, especially under drought conditions. This is work done by one of the person from Agiri here, Muhammad Saad. We looked at the role of some Hishak protein gene, and what we find, that's a Hishak protein gene, when you give stress for 4 hours, it shows, somehow it's hard to believe data, we did 4 times this experiment. We got almost a 2-fold increase in fresh weight and 3-fold increase in dry weight accumulation. It seemed like expressing Hishak protein gene, you could provide not only tolerance, but you enhance this growth prospects in plant. And we basically hypothesized that small HSP reduce inactivation of protein to overcome jamming of ubiquitin-based protein degradation. It's enhancing autophagy process in such a way that you delete the junk piling up in cells, and thereby cells are doing much better job of surviving. Other interesting phenotype of this was, when you put polyamine, you get a lot of flowering. And these flowers, not just flowers, these flowers actually set the fruits. So you can see this is a wild type, this is overexpression, this is antisense, this is post-suppression. Post-suppression has no effect like wild type, antisense has reduction, and overexpression show a lot of flower, and these flowers lead to a lot of fruit formation. This is again work done by Muhammad Saad Bella. So in summary, multiple phenotypes are attached with each gene. So you can't put one gene and see what one phenotype. There will be many different phenotypes come up. Polyamine regulate large number of processes in plants. More one days, more one finds actually. I have slide for thanking people, but I showed the picture of it where who did the work. So in consideration of time, I like to thank you, and I say I told you so, that calling the good stuff.